Hello, everyone. My name is Gerard Gatch, and I'll be the moderator for today's webinar on the topic of twin column capture LPLC technology and its practical application, or CaptureSMB, how does it work? This is a series of more than 10 webinars on multi-column chromatography for applications in the production of protein therapies. We understand that you have limited time and came here to hear the content, so we'll forego formal introductions of our company until the end of the webinar. Only a few housekeeping notes and we'll commence with the presentation. Today's seminar is approximately 30 minutes long, leaving. This presentation is focused on the basic mechanics and the economics of twin column continuous capture chromatography, but there's a lot more. This subject has many areas that we don't have time to explore in this 30 minutes. However, we invite you to explore our webinars recorded or planned that cover topics such as dynamic control, automation, process characterization, and end-user case studies. These are 10 to 20-minute presentations by subject matter experts. Today's presentation will go over twin column technology, how it works. We'll share a case study for a scale-up application, and then some economic aspects of continuous chromatography, and leave time at the end for Q&A. Today's presenter is Thomas Müller-Spath, who holds the position of Chief Technology Officer at YMC Chromacon based in Zurich, Switzerland. Thomas is inventor of more than 10 patents and has authored and co-authored more than 30 scientific articles and book chapters on continuous chromatography, which makes him ideal for this presentation. Thomas frequently presents at international conferences as speaker and also co-chairs workshops on continuous chromatography. Now I welcome Thomas.
Thank you, Gerard, for the kind introduction. Also, welcome from my side to this presentation about CaptureSMB, a twin column technology, how it works, and how it's applied in practice and what you can expect from it. So, first of all, let's have a look at where CaptureSMB is actually used in the downstream processing cascade of biologics. So here, this is shown for the example of a monoclonal antibody. So we'll focus today on CaptureSMB to be used at the first step, which is the capture step. So it's replacing a single column protein A affinity step in this case. Continuous countercurrent chromatography can be also applied in other stages of the downstream processing step. And also continuous chromatography, like the linking of two polishing steps, can also be applied further downstream. This will be covered in other webinars. So we have also webinars on integrated chromatography, and there are also webinars on continuous countercurrent bind elute chromatography for difficult purification. And this is called the MCSGP process, where our aim is to maintain a high yield. We've also had a webinar on this topic already last week for the purification of oligonucleotides and peptides. So today we want to talk about continuous capture chromatography. The motivation of using continuous capture chromatography is to improve the capacity utilization of the protein A affinity resin, which typically costs around 10 to 15,000 US dollars per liter. Now, with a typical size of protein A columns used in manufacturing of 100 to 250 liters, we end up with a packing worth of 1 to 2.5 million US dollars, assuming the lower price here. But in real life, the utilization is only 40 to 60% of this resin. And why is that so? This is because as we load the protein A column, we observe a diffuse breakthrough front as the antibody is occupying the resin binding sites. And this depends on the flow rate, on the titer of the antibody, also on resin properties. So let's say in a single column protein A operation, the column is filling up from the left to the right with monoclonal antibody, and you have this diffuse breakthrough front that is forming here. And at some point, antibody starts to break through from the column, and at that stage typically the loading is stopped to avoid losses of monoclonal antibody in the flow-through. So this means that we have quite some unused resin capacity, which is typically 40 to 60% of the total resin volume. That's a lot of capacity not utilized. Now that's the situation again as in single column chromatography. Now in continuous chromatography, what we do is we split the bed into two parts. So we have two columns which have a shorter bed height, and we then load these two columns in series. And we overload the first column on purpose beyond its dynamic capacity and capture whatever is breaking through on the second column. So this means that the capacity of the first column is fully utilized, and we do not have wasted capacity once we take this column aside and wash it and elute it and clean it. So what reduces resin lifetime is usually the cleaning of the column with caustic. So and if you take a look again at the single column, you see that once you elute this partially loaded column and you clean this partially loaded column, also the lifetime, so to say, of the resin that has not been utilized is decreased. So that portion of the resin that's not utilized also loses capacity, although it has never been used. Okay, so in CaptureSMB again, this improved resin utilization significantly decreases cost. We basically improve the amount of product that we're getting out per resin significantly, and for production columns, these savings in the case of protein A, that can be more than $500,000 for one column, set of columns. So the reward of using this improved capacity utilization again is also, in addition to a reduced resin cost, higher productivity, reduced buffer consumption, and smaller equipment and buffer tank footprint. So I will elaborate on these individual points on the following slides. And down here you can see the typical range of improvements that we expect to see when using continuous chromatography as opposed to single column chromatography. So this shows you productivity. This is for a titer of 5 grams per liter, increased by more than a factor of 2, capacity utilization increased by 50%, buffer consumption reduced by 50%. These are the numbers that are very typical with regards to improvement of use by using continuous chromatography. So let's have a closer look of how the process actually works. So we go into a refreshing animation now. So here we have our two affinity chromatography columns packed with the same material. Both are the same. The size of the columns should be reasonably similar. Typically we say, as a rule of thumb, the columns should be plus minus 10% the same height. So if we have 10 centimeter, one of the columns could be 11 or 9. We have means to balance and to account for that difference in the bed height. And this is by using UV-based process control. So in this first column now, we will start to load the first column, and we will see an internal concentration profile forming as the column is filling up with monoclonal antibody. So on the left column now, we're loading our cell culture harvest, our clarified harvest, and that clarified harvest is now filling up the first column, and what you see here in red is now supposed to be the monoclonal antibody that's filling up that column. So far so good. This is like in single column chromatography, but now we have the columns interconnected. So we continue to load beyond the point of dynamic breakthrough, and we transfer the overloaded material, basically the excess material, into the second column, where this material is reabsorbed. Now, following that interconnected loading step, we have an interconnected washing step. This step is important to remove any unbound material, to transfer any unbound material which is in the void volume of this column from the first column into the second column. I guess it's pretty clear that first column has all binding sites occupied with monoclonal antibody. So in the void volume between the resin particles here, there's still cell culture harvest, which does contain valuable antibody that we want to keep. So that antibody then is washed with a washing buffer into the second column, where there is still free capacity to absorb that antibody. Without that sequential washing step, now in the subsequent elution, we would lose the antibody that is in the void volume in between the particles. So we want to avoid that, but that antibody now has been transferred into the second column. So we're good to proceed with this column. We can have additional washing steps now, but the main purpose of this disconnected step here is to elute the product from the first column. So we now recover the product from the fully loaded first column. Again, you can see the first column now has been fully loaded. All the capacity has been utilized. So we can recover the product here, and at the same time we continue with feeding on the partially loaded second column. Now we do cleaning and re-equilibration on the first column, and at the same time we continue to load the second column. So this feed flow rate here on the second column that you see here is adjusted such that at the end of the cleaning and re-equilibration step, the antibody front on the second column is still within the bed height, so that no antibody is lost from the second column. So this means that, especially for a high titer, the feed flow rate here is slowed down, so that the completion of cleaning and re-equilibration and feeding here is synchronized and no antibody is lost. Okay, so now the first column has been re-equilibrated and is ready to take up a new antibody. And now we interconnect the two columns and continue with the loading on the second column beyond the point of breakthrough. So you can see now the columns are in opposite order. We basically are at the same situation as at the start of the process. Now just the columns are switched. So we continue to load the second column. We capture the breakthrough now on the first column that has been re-equilibrated. Then we wash the second column and transfer any material from in between the particles onto the first column. We elute the second column here, continue to feed the first column. So here we recover the product, continue to feed the first column. Now this is happening in parallel, product elution here and feeding on the first column. It's shown sequentially, but it's actually happening in parallel. Then cleaning re-equilibration of the second column and feeding of the first column. And then basically the cycle is completed and we can start over again. So as shown in the following slide, this is a cyclic process where that can go on in principle forever until all the starting material is consumed. So we have means to enter the cycle, so by a special dedicated startup step that takes into account that the columns are empty at the beginning of the process, we can introduce this step, and like this we enter into the cycle as if we had just come from within the cycle. So there's minimal interruption here, or let's say minimal disturbance, by using a dedicated startup step, which loads a little bit more, taking into account that the columns are empty initially. So then we can go on in this process again until all the starting material has been consumed, And to get out of this process again, we can choose either disconnected position. So at the end of the disconnected state here, one of the columns is washed, diluted, and cleaned. So that's ready to, well, to be stored essentially. And the other column now is partially loaded. And by just washing, diluting, and cleaning that column separately, we also get it to a state where it can be stored. So at the end of this shutdown step, both columns are stored and ready for, or they could be used for the next run. Okay, so I mentioned that feed flow rates in disconnected mode may differ from the feed flow rate in the interconnected mode, and this is dependent on titer and recovery and regeneration protocol. So here in the interconnected mode, we have a first feed flow rate, and then here we have a second feed flow rate that makes sure that these two steps are synchronized and completed at the same time here. Okay, so this process can be easily designed by single column breakthrough curve data, by taking single column breakthrough curve data and feeding it into a wizard that is a tool, a software tool that we supply together with our Condichrom equipment platform. So this tool then makes it easy to go from single column to CaptureSMB by making all the calculations for the flow rates, again based on the single column breakthrough curve that can be entered here and that can be then fitted using this wizard. Additional information is added, feed concentration, column dimensions, and the recovery and regeneration protocol. Now this tool also will predict the process performance based on the breakthrough curve. So even before having carried out a single run, you already get an idea of your expected performance from the CaptureSMB experiment. So this prediction is typically 10% accurate. So which means that the error is 10%. This can be very useful, for example, for facility fit calculations. So we have quite a track record of CaptureSMB established in the last years. So firstly, twin column multi-purpose systems are available for both lab scale and GMP scale. I think Gerard will mention a bit more about these systems later. So these are the Condichrom Cube and Twin series equipments. We have demonstrated scale-up at numerous users. Now this technology is in the clinical facility. There is a UV-based dynamic process control concept available. Virus clearance and carryover studies have been successfully established, and process characterization and validation concepts have been developed. All these points are important when it comes to manufacturing, and we have backed these individual points up by some nice publications in journals, biotechnology, bioengineering, and we have also one here in Bioprocess International, and I will talk about this publication a bit more now in greater detail because it has a very nice case study inside. Okay, so again, this is the case study I would like to present a little bit more in detail. It's from 2018, and it has been established in collaboration with BMS. So BMS has done most of the work, BMS in Devens, Massachusetts, and it's a great publication. So Now, all of the data on the following slides is taken from that presentations or publication initially. So the aim was to show if the Quantichrom or equipment is scalable, and also if the CaptureSMB process is scalable from benchtop scale to GMP scale. So firstly, we used the Quantichrom cube system, and then a CaptureSMB twin system from YMC Process Technologies, and they were run essentially one after the other with the same starting material and same resin, same protocols. And you can follow firstly the UV traces. You can see very nicely the concept, or you can follow the concept of CaptureSMB very nicely in these UV profiles here. So you see the breakthrough of antibody in these UV profiles here from one column to the other, while you see a constant impurity baseline that corresponds to unbound impurities that are breaking through from the columns. So whenever one column is loaded and antibody is breaking through from one column, you can see that from the other column there's no antibody breaking through, just impurities. So this is exactly the situation that we want to get. Down here, you can also see that breakthrough from one column and from the other column. Unfortunately, there's an offset here in the UV signal, so you cannot directly compare the UV baseline, but you get it that antibody is breaking through from one column, while there's no antibody breaking through from the other column at that very stage. So the protocol that was used for continuous capture and batch was almost identical. So actually, I'm showing here an abbreviated table in the paper. There's more detail. So first, the batch protocol, single column protocol, was translated directly into a continuous capture protocol by using exactly the same flow rates. So here you have 300 centimeter per hour linear flow rate for most of the steps except for the loading. And then in continuous capture, you see here the same steps with the same column volumes except for elution, where it was, in this case here, you see an optimized protocol. And you also see different flow rates here. So again, this already shows an optimized CaptureSMB protocol, and the main optimization you see here is due to the increased flow rate for recovery and regeneration. And I would like to point out why we have increased flow rate here for recovery and regeneration in the capture. That's the key element of this process. Before that, I would like to point out the increased load that we could achieve here with continuous capture, where we could go to 80 gram per liter versus 50 gram per liter in the single column run. Okay, so again, having a look at this slide that you already know, I would like to use this to explain why we can choose a higher flow rate in the CaptureSMB run than in the single column run. So in the single column run, we basically have a single bed height, let's say 20 centimeters, which we have to keep all the time. So this bed height we have for the loading, but also for recovery and regeneration. Now for CaptureSMB, we have a disconnected state where the columns are separated. So we only have 10 centimeter bed height here and 10 centimeter bed height there. So instead of 20 centimeters, we have 2 times 10 centimeters. So the bed height is only half compared to the interconnected mode. So this means we can increase the flow rate while remaining within the back pressure limits, right? Bed height is only half. So based on this formula that you're familiar with for the back pressure is a function of the bed height. So if the bed height is half, we can double the flow rate here and get to the same back pressure in theory. So this had been done in this study here. The flow rate had been increased. Theoretically, we could have gone up to 600 centimeter, but optimization, which is double the flow rate here. But we kept it a little bit lower here, just to get an idea of what the potential of optimization could be, and to be still on the safe side here. It was not fully exploited to its maximum capability. But flow rate can be increased, and this leads also to shorter processing time then. So the cycle in single column chromatography was 4.5 hours. Here it was just 2.5 hours. And yeah, so that definitely improved the productivity. And all of the higher load, obviously, that results in higher resin utilization and reduction of buffer consumption. Why is the buffer consumption reduced? It's because we are using the same amount of buffers for a load which is much higher, right? So the overall volume, let's say, stays the same, but we can load more antibody. So in terms of liters per gram of antibody, because the grams are more, we have a reduced buffer consumption. Okay, and again, here you have the summary showing the improvement of the productivity when going from batch to multi-column, twin-column chromatography. So this is for the twin scale-up system, and this is for the Quantichrome cube system, where we have a very similar productivity, and also for buffer consumption, we have very similar numbers between the twin system and the cube system. So this shows you that there's good scalability through from lab to GMP scale, and overall the improvement is large when it comes to single column chromatography comparison. Obviously, it is very important to check that you are in specification with the purity, and that the purity is the same between single column and multi-column chromatography. And here in this analysis, you can see actually that for CaptureSMB and single column chromatography, the purity in terms of host cell proteins, DNA, bleach protein A, high molecular weight, low molecular weight were very similar. Okay, let's have a look at a few economic aspects of CaptureSMB. So in CaptureSMB, there's a trade-off between capacity utilization and productivity. So maybe you have an idea from single column chromatography. The faster you want to flow through it, the more diffuse your breakthrough curve gets, and the lower your capacity utilization gets. So the same situation we have in multi-column chromatography. So if we go eternally slow, our capacity utilization will be very high. So if we have a very long residence time, our breakthrough curve will be very steep, our capacity utilization will be very high, but our productivity will be pretty low. So capacity utilization is actually defined as load divided by static capacity. So if that gets 100%, we have exploited the complete static capacity. Okay, so there's this trade-off between capacity utilization and productivity. So what does that mean? So if productivity goes down and capacity utilization goes up, we have a slower processing, our equipment is larger, but we have lower resin costs. If productivity goes up, we have faster processing. This means we can complete our job faster, or we have smaller columns. And we need more cycles per grams of product, and we have higher resin costs. So this trade-off I mentioned, that you know it for single column chromatography, it's also present in CaptureSMB, but it looks much better than for batch chromatography. So this modeling summary here shows that the trade-off for CaptureSMB is actually looks more rectangular than the trade-off for single column chromatography. So this means that even we can go up with productivity much higher and still maintain a decent capacity utilization. In single column chromatography, we go down quickly or steadily. Now in multi-column chromatography, we can stay up at a higher level for a longer period of time, or let's say for increasing productivities, we can stay up at a decent capacity for much longer. Okay, so on this previous slide, you have also seen a comparison with other multi-column processes. They all share the same feature that they can reach high capacity utilization. In terms of productivity, there's not a lot of difference between 2-column, 3- and 4-column processes. However, when it comes to complexity, obviously there's a large advantage of a twin-column process that only uses 2 columns, because it also has only 2 pumps and a much smaller number of valves. So this becomes particularly important when you think about scale-up. On the bench scale, it's not such a significant aspect, but complexity does become very important when it comes to manufacturing, because increased complexity means increased downtime or risk of downtime and increased risk of implementation and validation. So with a 2-column process, it's the simplest multi-column process, thus also complexity and risk are the lowest. Now, for putting some numbers on the savings that you can estimate, that you can get by CaptureSMB. So we have here a scenario, first scenario for 1 gram per liter titer. Here we can expect savings of protein A resin material in the range of around $900,000 US, 400 kilograms antibody produced. So you see here we compare either the same processing time or the same resin costs, which means the same overall column volume. So it's due to the advantages of CaptureSMB, either you save a lot of time or you save a lot of resin costs. So of course you can imagine scenarios being somewhere in between. This is for a 1 gram per liter titer. Now for a 5 gram per liter titer, the improvements are still significant, although a little bit smaller. So we still save a couple of $100,000 US, 400 kilograms, and we save, again, a lot of time. Okay, so yes, summarizing again the advantages of this technology, I hope I could convey the major points here. So significant productivity gains can be achieved through CaptureSMB. This is due to the improved resin capacity utilization and the faster loading. So we can afford having a shallower breakthrough curve in CaptureSMB, and we can therefore achieve faster loading. We can achieve a shorter processing time because we can increase the flow rates. We have reduced buffer consumption. And so these are the advantages, and also this study, in various studies, I showed one of them. There will be more coming up in other webinars. We showed that process scale-up can be achieved from bench scale to process scale. And we have good reproducibility, and we also have a simple 2-column approach which accelerates implementation and minimizes the validation risk. So with this, I would like to thank you for listening, and then I would like to hand it over for Gerard for some concluding remarks before we move into the Q&A session.
Thank you very much, Thomas. That was an excellent presentation, really. So attendees, please continue to submit your questions now. We do have a good number of them, but we certainly have time to answer more. You can submit those by entering your question in the Q&A box, which is in the lower part of your screen. You'll be receiving an email from us that will give you some reference to the materials that Thomas referenced, such as some of the papers that you might want to look into in the coming days. This presentation was sponsored by YMC Corporation. You may know YMC as a leader in chromatography, producing bench and prep scale HPLC and LPLC systems, packing resins, columns since 1980. YMC is manufacturing in offices in over 10 countries. And so reach out to us if you want to look into more of the products or feasibility studies. Now, what you may not know is that YMC recently purchased both Chromacon and the former Lewa Process Technologies, who were partners in developing both bench scale and GMP scale systems. So this added significantly to YMC's 40-year history and now puts YMC into a leadership position in the industry in the emerging area of multi-column chromatography. So the result of these acquisitions is to provide a seamless platform, as Thomas alluded to, from the bench to GMP scale. And there's so much more to this technology. Just as a reminder again, there are a lot of other aspects that we didn't have time to touch on today that talk about the tools and other insights, automation, characterization, further case studies, which will be available to you in many webinars. I'd like to bring Thomas back for the Q&A. And Thomas, we do have a good number of questions. Thank you for submitting them. Keep doing that. Let's see, let's go through and take a look. Okay, first question. What happens if the titer changes while running this process? I don't think you covered that.
Yeah, I mentioned different titers, but I didn't mention what happens if the titer changes. So this could happen during, for example, a perfusion process. So for that case, so we have two options. So either we design the process more conservatively from the beginning using our breakthrough curve, or second, we can use the dynamic process control feature that I also briefly mentioned to handle that. So this will be part of a separate webinar. It's called the AutoMap UV-based process control, and that is monitoring the UV signals as they elute from the column, and then it takes action based on the UV signal rising as the antibody is breaking through. So if you have, let's say, higher titer, your antibody signal would rise earlier because your column fills up earlier. So then the controller would take action and stop the loading earlier.
Thank you. Next question, if I can interpret this, do I need continuous upstream to run CaptureSMB?
Yeah, so that's a little bit linked to the previous question maybe. So no, the answer is CaptureSMB works with both continuous upstream, so perfusion, and fed batch processing, so fed batch fermentation. So I showed two cases, right? 1 gram per liter titer and 5 gram per liter. So 1 gram would be representative of perfusion, and 5 gram per liter, for sure, that's fed batch. So it works in both cases, and in both cases you get these advantages of improved productivity, improved resin costs, lower buffer consumption. So there are some companies who really say, okay, we don't have the expertise in perfusion. We are a fed batch company, so those companies can also benefit from the use of continuous downstream processing.
Very good. Does CaptureSMB work only with protein A?
Yeah, so it's a general process principle that works with all bind dilute materials. Obviously, it makes sense most where resins are expensive, where the lost resin capacity really hurts, and that is for affinity materials like protein A. But there are also other, even more expensive affinity materials where CaptureSMB would make even more sense. So, for example, for affinity materials used in monoclonal antibody fragment purification, there the savings can, in terms of US dollars per antibody or per produced product, can be even larger than with protein A.
And I think this question segues quite nicely with that answer. Does CaptureSMB also work in the polishing stages?
Yeah, so again, it does work there, but because you're using mainly, let's say, more cost-effective resins there, like ion exchange resins or let's say hydrophobic interaction resins, where the resin costs are not so high, it may not be the primary place where you want to make or realize resin costs improvements. So these costs are not so significant there. In addition, also CaptureSMB is very good with affinity materials, so which are digital, right? So there's the protein A or the affinity material captures the molecule of interest, the antibody or antibody fragment, whatever, and lets everything else pass through. Now, in polishing steps, you usually want to purify the product from product-related impurities, such as aggregates. So CaptureSMB could not purify, you cannot separate aggregates because they're product-related and they're binding also to the affinity matrix. So in that case, another process may be more applicable, like MCSGP for the removal of product-related impurities.
Is there a difference in how I should develop my process if I want to use this CaptureSMB system?
Yeah, generally not. So we've seen in the study that we have been using the same steps in terms of washing, elution. So we should start with the same buffers, trying the same buffers, and chances are that they work well also with CaptureSMB.
Here's one I think I can handle. Has this technology been scaled up to GMP production? And I believe you did allude to papers that have been co-authored with end users demonstrating scale up. And I know our facilities have been able to have standard models all the way up to 20 liter a minute using CaptureSMB. Okay, next question. Where do you see the highest hurdle when implementing this into a GMP production scheme?
Well, I see the highest hurdle is basically that it's a new technology. So there's little experience in the industry so far. I mean, a number of important aspects have been covered. So like the virus clearance, the validation, but yet no, there's no drug on the market that has been produced using this technology. So we are very optimistic that it can be done, because I mean, we're using the same resins, we're using the same buffers. The studies that I just mentioned, virus clearance, carryover, have been good. And so far we don't see any red flag. Also, we know from the past that multi-column technology has been successfully scaled up. That's for simulated moving bed chromatography for the case of chiral molecules. So there's a track record of that being scaled up. So and generally chromatography is a very well scalable technology. So we're optimistic that it can be done. Yeah.
Here's an interesting one. Would this technology work with membrane chromatography?
Yes, in fact, it does. So we've also done it. So we've used the CaptureSMB process with membranes from two vendors in lab scale format, and it also works very nicely. So especially with membranes which usually have a lower capacity or dynamic capacity than resins, it is important to squeeze out any gram per liter of loading capacity that is there. So yes, it works well, and it can improve also membrane chromatography.
Okay, I want to be respectful of everyone's time from the attendees. So I'll look through and pick out one or two more questions, and then we'll do a wrap up. Would you realize greater capacity utilization with more than 2 columns?
So this it depends on which kind of affinity step is used. So in the case of monoclonal antibodies and the current resins that are on the market, the profile of the monoclonal antibody fits very well into 2 columns of the typical bed height, let's say of 10 centimeters, right? So this means that when the first column is fully loaded, the rest of the profile that is broken through fits into the second column. So there is no use or no advantage of using another column that's now in this sequential, in this interconnected loading train. So it would just mean that not only the first column is filled up, but also the second column is completely filled up. So it doesn't give you any advantage. So, and in fact, in most processes that have been presented recently, you find that for monoclonal antibodies, only 2 columns are present in the loading zone, and this means that the load, the capacity utilization, buffer consumption, and product concentration will be the same for all these processes, no matter if they have 2, 3, or 4 columns.
Thank you, and we'll take one final question, but I'll tell everybody that there's a number of other questions. We'll try and issue all of these questions and respond in writing with maybe some format of FAQ so that everyone's question can be attended to. So final question, how can CaptureSMB handle column degradation? I'm assuming they mean by the resin degradation over time.
Yeah, so that's a quick answer this time. So because I mentioned it, I think earlier that the difference in terms of bed height can be handled by the UV-based dynamic process control, and likewise is the column degradation be handled by UV-based dynamic process control. And I would like to refer to the talk on the AutoMap, which we have also scheduled in one of the next weeks.
Very good. Again, thank you, Thomas. Great presentation. And to everyone who attended, thank you for your questions, your participation today. For those who have immediate application, reach out to us at info@ymcpt.com. That's info@ymcpt.com. And in a short while, a couple days, you'll receive a link to an archived version of this presentation and a link to the associated reference documents that Thomas referred to. Feel free to share these with your colleagues. And as a reminder, this is one in a series of webinars, all of which you can find posted at biowebinar.com or our resource page on our website. We'll end the session now. On behalf of Thomas and myself, thank you and have a great day. Bye, everyone.